I'm Melanie Foster. I'm going to talk to you about the training and validation plan that my colleagues at ILS and I used to prepare for an extended one-generation reproductive toxicology study, covering all the phases of the reproductive cycle and development, starting with exposure to the parents pre-mating, through mating, gestation, and lactation, and through the life of the pups. The parents and the offspring are assessed for general and reproductive toxicity, and in addition, the offspring are allocated to 5 different cohorts. 1A and 1B are for reproductive and developmental endpoints. 2A and 2B are to assess developmental neurotoxicity, both at weaning and at adulthood, and cohort 3 is to assess developmental immunotoxicity. These studies have many endpoints that are not found on general toxicology studies, such as estrus cyclicity, developmental landmarks in offspring, auditory startle, motor activity, and functional observational battery in the offspring, perfusion fixation for histopathology, including brain morphometry. Also, the reproductive system is examined in more details, such as counting ovarian follicles and implantation sites. Sperm motility, concentration, and morphology are assessed. And for immunotoxicity, there's an analysis of the splenic lymphocyte subpopulation and a T-cell dependent antibody response assay. Our plan had 3 steps. First, equipment validation. Next, method development and training, and third, a performance validation using positive control substances. Equipment validation was done using validation scripts to conduct installation, operational, and performance qualifications to comply with the FDA CFR Part 11 for computer-generated data. We validated multiple systems, including photobeam activity system, acoustic startle, grip strength gauge, thermometer, and the sperm analysis system. And this was on the hardware as well as the software. An example of method development was for the TDAR assay. We did a stability study and determined that we could use the same sheep red blood cell preparation for up to 3 days. For the functional observational battery, we trained research assistants in pairs to facilitate standardization, and we began with in-classroom training with video, then did multiple sessions with control animals until they were comfortable with the assessments, followed that with sessions with positive control treated animals, and then finally a blinded certification test using positive control treated rats. We used corn oil, DDT, and carbaryl for our positive controls. The results of our certification test showed 98% concordance between the 2 trainees and the trainer, as can be seen on this graph of hind limb foot splay showing 3 different individuals assessing animals. For motor activity validation, we used saline, chlorpromazine, and amphetamine as our positive control substances. And as you can see, the blue line is saline, the green line is amphetamine, and the red line is chlorpromazine. And saline-treated animals, they have a high activity at the beginning of the session, which decreases. Amphetamine-treated animals did not decrease in their activity, and chlorpromazine animals stayed low for the whole session. For neuropathology, not only is the standard neuropathology assessment required, but also brain morphometry, which requires homologous sections across the dose groups and measurements of various structures within those sections. There's also extensive reproductive pathology, including staining the uterus for implantation sites at necropsy, H&E for further evaluation, evaluating vaginal smears for estrus cycle, counting the ovarian follicles, evaluating sperm motility, concentration, and morphology. These studies have many different parts, and it requires a collaboration in order to get all the components validated, but a comprehensive validation study is essential in order to be able to confidently carry out the full extended one-generation reproductive toxicology studies. I want to thank everyone at ILS that helped in getting this study completed.